For part two. I bet you are. Yeah. All right. Yeah. So that thing we just looked at, that was how B cells rearrange their DNA to make antibodies. You want to bet how different the process is for making T cells? Yeah. Don't bet much because it's not very different. Not very different at all. So let's go. In T cells, instead of having a light chain and a heavy chain, you have an alpha chain and a beta chain. And the alpha chain works like a light chain. It has variable, join, and constant regions. The heavy chain, the beta chain works like a heavy chain. It has variable, diversity, variable, diversity, join, and constant regions. And they're organized about like this. So there's a set of, a diversity region will have a certain number of join regions after it. And if the recombinase, if RAG1 and 2 select this diversity region as the one that they're gonna commit to, that's fine, and they'll pick one of these join regions, usually that comes right after it, and hook it up to this constant region that comes right after that. So usually this constant region will go with one of these joins, which will go with this exact D region, which will go with some random V region. The only other difference in the T cell genetic makeup is that there's an extra exon. And this is the leader exon. This is L1 and 2 and 3. There's one leader exon per variable region, and they're two exons. So they're going to be spliced together, and the leader doesn't encode anything. It's just the 5' untranslated region of the messenger RNA that's going to go out and make these. So it's a thing that's there, but it's not a thing that affects the actual coding sequence of the of the T cell receptor. All right, the T cell receptor. So you're gonna have recombination to make the light chain, which is the alpha, and you're gonna have recombination to make the heavy chain, which is the beta, and basically the same thing. First step is going to be to get the D and J together. Second step is going to be to get the V and DJ, or just V and J together. Third step is going to be to get the VJ and C, or VDJ and C together. Yeah, just like you'd imagine, right? Yeah, just like you would imagine. All right, and so just like in antibodies, they're those same invariant recombination signal sequences, the 12 nucleotide and the 23 nucleotide. And so those are there in, I believe, exact form every single time. And so really, if you think about it in evolutionary terms, all these variable domains are just like a gene duplication. You probably started out with one, and then it got duplicated, and so now you have two, and then somehow those got duplicated, and maybe you have four, and then maybe three out of those four got duplicated, and now you're up to 7, like that, over and over and over again throughout history. And each time, apparently it didn't hurt the creature that had this horrible gene duplication, and may have even helped them. And then over time, those variable domains started to wander. They got a little more different from each other. And we ended up with the diversity that we have today. Yeah. So that's the idea. It works the same in gamma delta T cells. In gamma delta T cells, there's a heavy chain and there's a light chain as well. I do not remember. Actually, I do remember. So the delta is the heavy chain, and the gamma is the light chain. So alpha light, big bad beta, gamma light, ding dong delta. I don't know. It's just delta's big. Yeah, delta's a monster. Okay, fine. So if we're talking T cells, between your alpha betas and your different immunoglobulin types, heavy and light chains, here we go. This is the total number of pairs. So if you have a particular set of variable genes hooked up in some way to diversity and join and constant regions, what you end up with are, around 10 to the 13th potential antibodies that you can make. And around 10 to the 18th potential T cell receptors that you can make, just the alpha betas. Gamma deltas, you can make a large number as well. Think how many cells you have in the body. It's a much smaller number. You can look up the number. I think it's somewhere around, I don't know, I'm guessing like 10 to the 12th. But there are basically more possible combinations of T cells than you'll have cells in your body. And your body does not consist solely of T cells. Yeah. No matter what your mother might have told you. So, basically, there are combinations of exons that you could make that might even totally be helpful in fighting whatever it is your body is fighting right now. And you may never make them in your entire lifetime. They may be sitting there in your DNA unused. Yeah. Unloved. Yeah. Unexpressed. Yeah. And they just need to be expressed. That's immunology. That's why immunology is hard. There are aspects where not everybody can make a particular antibody because there is diversity. Some people are just missing a particular variable chain. And there are aspects where it's there, but you may not use it. You may not be able to access it. And so it's a race. Can you sort through this? Like, it's not infinite, but it's large enough that it's a pain to sort through, and you may never get all the way through it. Can you sort through this box of tools and come up with the thing or things that work in time before the body dies from whatever is beating it up at the moment? That's the race. That's the uncertainty of immunology. This is why we have problems, and this is why even when you give everybody the same vaccine, they've still got to go through this entire process for every single antibody producing cell and every single T cell receptor containing cell that they're going to make. Everything they can do is guided by a random process. And there's some solutions that they can't even make and other solutions that they can make, but may never make. Okay, that's enough harping on that, but I think it's interesting. So there we go. We don't even need to talk about those. We're just looking at so here's an MHC molecule on the bottom, and so this is showing where the different loops, complementarity determining regions of a T cell receptor will bind, and what do you know, they are all going to be arrayed in a place where they can interact with the peptide. So there are some amino acids on the peptide that's being displayed that are being held by the MHC, and all the other amino acids are fair game for these little hypervariable loops to try to bind. And so, can you get a match in time? The race is on. And fine, the process works about the same. You loop these things out. It's still RAG1 and RAG2 doing the job. Not a lot more to say than that. The thing that we want to cover, though, is different types of antibodies. And so antibodies of these different classes, like IgA, IgM, IgE, can have exactly the same, will generally have exactly the same variable region. So weirdly, the variable region on an antibody ends up being constant once it's made. And the thing that changes is the constant region. Yeah, did that mess with your mind? I don't know. I figured it messed with somebody's mind when they were coming up with these terms and then realized, oh, wow, they're all kind of backwards from this particular point of view. So every antibody starts out as an IgM. That is the first thing. And an IgM with an extra transmembrane region on it is the initial B cell receptor. That is then going to get converted to an IgD. And it always goes from M into D, and M can then be released. But the cell, the B cell, as it develops, is going to go through different constant regions. So once it's selected, V, D, and J to join together to make like one super duper exon, it's then going to experiment with different C's. And each time, if it makes a constant region and then gets a signal back from some other cell, like a antigen presenting cell or a helper T cell, for example, then it's going to say, well done, and it's going to maybe move on to the next. So M is always first, D is always second. After that, it's kind of variable. We used to think that they went in exactly this order. So 1, 2, 3, 4. So M, D, gamma 3, gamma 1, gamma 2b, gamma 2a, then epsilon, then alpha in a mouse, and in a human, MD, gamma 3, gamma 1, and then the psi gene is a pseudogene. It does not work anymore, but it used to be a copied version of the epsilon. It was like a duplicate of the epsilon. And then alpha 1, and then gamma 2, gamma 4, epsilon, and alpha 2. That would be the order. But once it gets past delta, it can actually jump around. So it can, this is the current understanding anyway of how this works. It can jump from delta straight to alpha 1 potentially, or go straight to gamma 2, and then it could skip over gamma 4 and go to IgE, and it could skip over gamma 2 and IgE, go to gamma 4, and then go to alpha, whatever. Yeah, you can imagine the combinations. You don't need me to say them because that doesn't help anything. And if you look, these constant regions contribute a lot to the antibody. So they're a big component of the antibody's size. These, like IgM and IgE, actually have 4 immunoglobulin-like domains that are part of the constant region of the heavy chain. For the others, there are only 3 immunoglobulin-like domains. You notice some of these will make one disulfide bridge, but way down here between those domains. Others will make it up here in this little sort of neck region. Some will make 2, some will make 1. And the little hexagons, little gray hexagons on the side, these are all glycosylation points. So you get sugar molecules attached. And there are proteins that can bind to the sugar molecules. The sugar molecules also prevent other proteins from binding. Everything gets very complex at this point, and it's better to just focus on specific things that we understand and know happen. But there is definitely still room for research in this area. There are interactions that happen out there that we probably do not yet know. And whatever, become an immunologist, see if you can figure them out. Yeah, you'll be doing humanity a great service. And future generations will have to learn about your thing in their textbook. Yeah, maybe that feels good. I don't know. I have no idea. Yeah, that's the idea. So this process is called class switching. So you'll start as an IgM class and then an IgD class. And then you'll go GEA, or GAE if we're in human terms. But the G, A, and E can all happen kind of in whatever order. It's going to keep moving basically from left to right as they are on your screen, but it may miss out some of the intermediates. Yeah, it's not a perfect thing where it has to do 1, 2, 3, 4, 5. It can do 1, 2, 5. All right, fine. Yeah, they are differentiated by their constant regions. And so some of these are actually very large. Like look at the IgM constant region. Or rather, the full immunoglobulin is massive for IgM because there's 5 of them that are going to get joined up together. And each of these versions of an antibody has different properties. So shown here is how well they activate complement. And it's not a thing that's worth committing to memory, I don't think, but look at the differences. So you make a lot of IgM early on, and that's great because early on, that's a good time when you want to blow things up with the complement pathway. And it just so happens that IgM is really good at activating classical complement pathway you'll have to remember all the subunits. I think C1q might be toward the start of that. Yeah, and then they're going to have different degrees of ability to bind to different things. So macrophage and other phagocyte, like neutrophils, have some phagocyte function, which just means they can eat things. They eat things, and then they can display a little bit of whatever they eat. It's exactly like a kid, eating a bite of hamburger or something, and then going, yeah, and showing you what he just mashed up. That's That's what antigen presenting cells do. They eat stuff and they show you what's on their tongue, basically. They actually decorate themselves in little bits of it, which is something that babies definitely like to do. So, yeah, just like babies, antigen presenting cells. Who knew? The thing I think to focus on here is look at IgE. So it's kind of bad at everything. It's not great at classical complement activation. It's okay if you stick an IgE to something. You can you eat it better with a macrophage. Macrophage will consume the thing a little bit more avidly. But the thing they're amazing at is binding to the IgE receptors that you find on mast cells and basophils. And when that happens, that can be a powerful way to fight some parasitic infections. But more commonly, in this mostly parasite-free age in which we live, that tends to be where you get allergy. So you'll have pollen or something. You'll make an Ig, G does nothing. IgM does nothing against it. And then finally you make an IgE against pollen. You're just like, I don't know what else to do. Let's make one. And then, the IgE finds a little bit of that pollen that's somehow made it inside your tissues, and it's going to react, and it's going to really fire off a mast cell or a basophil. Those are highly pro-inflammatory, and so you're just getting a localized inflammatory response to IgE that's bound to something. And that's a large component, not the only component, but a big component of what allergy actually is. So there you go. We will cover that in more detail. There's a whole section in, I think, chapter 14, but we'll get to it. Yes. So how does this whole thing work? So you have your IgM, and you're going to start out with an antibody that has the nice little IgM on it. And that same transcript can actually be processed in different ways. So you make IgM and D kind of together. You'll make a pre-messenger RNA that could be used to make IgM, or it could be used to make IgD, and so it'll just combine these two different chains on, and the process is determined by splicing, and either one can work. You do tend to get IgM first, but you'll get a bit of IgD once you have started making IgM. Yes. The other important thing, so what's the difference between a B cell receptor and an antibody? There we go. And the answer is transmembrane region. So this is also something that is going to be determined here. So we're going to have heavy chain splicing differences. And so you're going to have some of these genes. So this is a pre-mRNA in both cases. If we're going to make the secreted IgM, what it does is it joins up these regions, and it puts this little SC region at the end. And if the SC region is at the end, that's the soluble C terminus, and that basically just stops the antibody, truncates it. And so it's at the level of mRNA splicing that we're going to determine whether we make a secreted soluble IgM, or the other alternative is we don't put that SC, the soluble carboxy terminus region in, secretory carboxy terminus region in, rather. We're going to put in, there is actually a membrane region. It's two exons, the M1 and the M2, and those are going to get joined together. And if we make that, then we'll make an antibody molecule with a little transmembrane on the end. And so they're both there in the same messenger RNA. So if the cell is making some IgM, it's also going to make some B cell receptor at the same time. And you saw from the bonus quiz how that gets used, and I believe we come back and cover that in a little more detail in a later section. So there you go. I think that's about it. Oh yeah, the last thing. So remember, you get some antibodies that are just, one set of two heavy chains and two light chains. You get other antibodies that are two of those, like the secretory IgA molecule, or the pentameric IgM molecule with 5 of these there. The thing that joins them together is an extra little chain called the J chain. It's different than the J exon. That's a joining exon. This is a joining. It's like a separate little protein that is made that actually sticks all of these together. It's like a little glue protein that brings the 5 antibodies together or brings the 2 antibodies together. Yeah. And then allows them to, instead of disulfide bonding to themselves, they'll disulfide bond to each other. So that's the thing that IgM can do. And IgA, shown here, can disulfide bond to the little J chain. So there you go. That's antibodies, and that's enough for Chapter 5. All right, thanks very much.